Dr. Camille Desjonquères. She studies how the biotic and abiotic environment affects acoustic signaling in animals, and she's particularly interested in the dynamics of acoustic communities and the effects of social interactions on the acoustic signals, and is currently a postdoctoral fellow in Wilfred Thruer's team at the University Grenoble Alpes, where she is setting up a network of acoustic recorders along elevational gradients in the Alps and the Pyrenees to monitor mountain environments. So I shall pass you over to Camille.
Hello, everyone. I'm very happy to be here and to present the work, some of the work I've done as a PhD student and as a postdoc in France and in the US. And the talk will be about hidden acoustics, monitoring underwater, and plant-borne insect sounds. So I want to first thank my collaborators on these projects, from conceptualizing to leading fieldwork to analyzing data and writing manuscripts. They have been super helpful and always ready to discuss ideas. So it's been a pleasure to work with them. I also want to thank the funding agencies and the institutions in which I have worked for these projects. So if you're thinking about insect sounds, you're probably thinking a cicada or a cricket, something like this. Or something like this. And of course, these are sounds that you can hear from insects, but there is a huge diversity that is unavailable to the human ear without any equipment. For example, aquatic insects produce sounds that can sound like this. Or plant-borne vibrations, such as something like this. So indeed, in 2005, Rodriguez and Cockcroft showed that actually what we know as insect sounds, cicadas and crickets, Orthoptera and Orthoptera, are not the majority, of insect sounds. Indeed, most of the sounds are substrate-borne vibrations. And even in animals, insects that we know produce sounds, and we think we know their acoustic communication system, we are discovering that they also have the ability to hear, to sense vibrations, and sometimes we discover that they actually use those vibrations for their communication. So there is a huge so many insect sounds are not audible to directly to the human ear without any equipment. And these sounds are hugely understudied. So in this presentation, I want to show you a few of the solutions that we have to study those insects, and what the type of studies that we can do with these insect sounds. So first, I will talk about underwater insect sounds, and then I will talk about substrate-borne sounds. So during my PhD, I wanted to know what are the animals that produce sounds underwater. And so I looked, I recorded in different forms, and I realized that it can be quite the cacophony. And so I conducted a literature review and found that there are three groups of animals that produce sounds on the water: amphibians, fish, and arthropods. And among those, here are some examples. So an insect, an amphibian, that which is the Discoglossus pictus, which produces sound really completely submerged underwater. And the sound of a fish, it's a short grunt, so you have to listen for it. And so I conducted this literature research on animals in France, and I found that 271 species produce sounds underwater in freshwater in France, and that there is a huge gap in terms of the description of those sound production. So out of those 271 species, there is only 90 species that are for which the sounds are properly described. And then if you look at the total numbers, you actually find out that 89% of the sound producers are aquatic arthropods. And so in this group, there is only 35% of the species that are described in terms of sound production. And we conducted another literature search with Jack Brinhard, and we found that in terms of number of studies on freshwater bioacoustics, there is a huge discrepancy between the number of species that are producing sounds in different groups, especially in arthropods, and the number of species studies that are published on the subject. So there is clearly a lack of study in this area. One thing that is not really related to the talk of today, but I think is cool, so I'm still including it, is when I was doing my PhD, we discovered that plants produce sounds on the water when they're photosynthesizing. And so just to show you what a plant sound is like underwater. So we found that there are many taxa that produce sounds. However, there is no reference database. So there is a huge lack of study. So one thing we did was to study one species to describe better their activity. And so we looked into Micronecta schultzi, which is an insect, an aquatic bird that measures about 2.5 millimeters. And that produces a sound that is about 120, up to 120 decibels. So it's a very loud insect. And it's actually the record holder for the loudest animal compared to its size. So very loud insects that live in this kind of pond. So we recorded this pond with hydrophones so underwater microphones that were connected to SM2 recorders. So it's the predecessor of the sound meter from Wildlife Acoustics. And so we set up a schedule of recording 1 minute every 15 minutes for 3 weeks, continuously during the day. And we set up 12 recorders, 12 hydrophones within this pond, where there was a noise source which mimicked the noise of a boat. And so we had 3 weeks. First week we just studied the free activity cycle of this species. The second week we could control the noise, so we set up the noise to play during the maximum activity of this species. And then the third week, we stopped the noise completely and saw what was the dynamic of activity during that week. And so to study the activity of the species, we looked at the sound that the species is producing. And if you look at it in the lab, of course, you will see clear temporal patterns. So you have already played that sound to you earlier. Here is what it sounds like. But in a natural environment, this insect is chorusing. They're many individuals are producing the sound at the same time. And so what you get instead of clear temporal patterns are just a frequency band that is quite that shows up. And the other thing that's striking when you're recording in the wild is that there are other species that are producing sound at the same time. And so what we did was to use the amount of amplitude within that frequency band, the species frequency band. And so we used the mean spectrum of the recordings and quantified the amplitude between 7 and 12 kilohertz, which are more or less the limits of the sound the frequency of the species. And so we first validated this method by comparing the index with listening score, which we used to annotate 300 recordings, sorry. And so we see a clear relationship between the acoustic activity that we listen to and our acoustic activity index. So then we use that acoustic activity index to quantify the daily cycles of activity of Micronecta schultzi. And so we found that there were activity peaks just before dawn and around mid-morning. So here you have the acoustic activity over time during a week, and the gray area is nighttime and the white area is daytime. The second thing we found was that the noise treatment had an effect on the acoustic activity of the species. It made the species increase their acoustic activity as well as delay their peak of activity. And that effect, both the delay and the amount of activity or the intensity of activity, did not go back to normal after the treatment. So with this method of recording with hydrophones, we were able to study daily activity of an aquatic insect and quantify the effect of a disturbance on this activity. This helped us to get better knowledge on the signaling of this aquatic insect. So the second part of this talk will be about substrate-borne sounds or vibrations made by a terrestrial bug called Enchenopa binotata. And these are of the family Membracidae. They're North American species, part of a complex of different species. And so one thing that's very interesting about them is that the male and the female tend to duet with each other. And so the females will, receptive females will only respond to males that they find attractive. So that gives us a very good method to assess female preferences. And so here is what a duet sounds like. Another interesting aspect of this species is that as juveniles, they tend to live on different aggregations, in aggregation. And there are two species in Wisconsin where I was working. One that is more black with a white stripe as juveniles. As adults, they look exactly the same. And one that is more brown grayish, and they can be found in nature together. And as juveniles, they also interact with each other and have a specific repertoire which is different from the adult repertoire. So here are some of the sounds that they make as juveniles. So here we were interested in assessing the effect of social interaction that they experienced in aggregation on the signals and preferences of adults. And so what we did was to record adults with laser vibrometry, which is one of the methods you can use to record plant-borne vibrations. So you use a laser that you point at the plant on which the insects are signaling, and that allows you to record the vibration. We also use vibrational playbacks. So we use synthetic signals for which we varied the frequency. And that allowed us to ask questionnaires to the females using those those synthetic signals. So we could vary the frequency and count the number of responses of the females to different males. So that gave us a curve of the number of responses in function of the frequency of the male. And so we were able to assess the female's peak preference, so the frequency at which female response is maximal, and so that gave us their preference. So we manipulated the social experience of young tree hoppers in two different ways. First, by isolating individuals, which is not an so-called natural condition for them, because they usually grow up in aggregations of 20 to 40 individuals. And the second manipulation we applied was to mix the two different species. So we had either only one species on our plants, or two species, but we kept for this experiment, we kept the density of species constant. So we had an intraspecific part and an interspecific part. And so what we found in the intraspecific part was that isolated individuals tend to have a lower frequency preference for lower frequencies. At the interspecific level, we found an effect of the treatment on high frequency species, where the high frequency species tended to go away from the low frequency species when they were mixed when they were in the mixed aggregation. So this suggests that this social plasticity response could help in enhancing species divergence. So with this effect, with the study on the effect of social interactions, we were able to monitor signals and preferences emitted by a substrate-borne producing insect. vibration producing insects. We were able to assess the effect of social interactions within this species, and to show the potential of interactions to influence the evolution of signals and preferences. And we were able to show the effect of sexual selection on these signals. So just as a perspective, I just got a permanent position as a CNRS researcher here in France at the University of Grenoble. And so I'm going to be setting up an acoustic monitoring network in the Alps, where we are recording along elevational gradients that are targeting a diversity of environmental conditions along elevational gradients. And so this project is part of an observatory called Enchant, where a lot of ecological data is collected, the vegetation structure, the pedology, the climate, there's insect trapping, camera traps, soil DNA. So a lot of ecological data that we can combine to the acoustics data. And so my project within this observatory is to set up acoustic groups and relate those acoustic groups to environmental data and look at how they interact with each other. And so with that, formulate spatial temporal models for the presence, absence, and abundance of acoustic groups. And that will allow me to understand acoustic niches, to map acoustic diversity, to predict global change, and to guide conservation strategies for the future. So I'm very excited about this future work. So I want to thank you for your attention, and I'm happy to take any questions if there are any. Thank you.
Thank you so much, Camille. So I've got cold, that's why I put this on. Amazing talk. There's questions coming in left, right, and center. I've got loads in my head as well because it was so interesting, especially with the tree hoppers creating such low frequency sound. I wouldn't have expected that at all. Is the reason for that just because it travels further? Is there a reason for this low frequency? Is it less energy? Or do you not know yet?
That's a very good question, actually. Is thinking about this, usually there is a strong relationship between frequency and size, and so insects are quite limited in the frequency, the lower end of the frequency that they can emit. But it looks like in plant-borne vibrations they can emit much lower frequencies. And we don't really know why. There probably isn't so much, constraints on what they can do, and we're not sure how to quantify actually how much energy they have to put in to emit those sounds. So that's something we were interested in, really interested to look at. Yeah, and I see. As a colleague, Jerne, saying that the vibrations obey to quite a different set of physical rules, which is absolutely true. Yeah, airborne is much harder to emit. So yeah.
Anyway, I'll do proper questions now as opposed to ones that I want to ask you. So how do you record the substrate sounds made by the tree hoppers, and how are they making those sounds?
So the tree hoppers, so how we record them is using laser vibrometers. So they're just lasers which use Doppler to assess the emission of sound, or just to measure vibrations. And then the second question, how they emit those sounds, they actually contract muscles that, you know, they contract those muscles, and that makes the plant vibrate.
Excellent, excellent. So interesting. Thanks for all those sounds. So this is quite a good one. How do aquatic plants make sounds?
So we're not quite sure yet, but we think that so when they're photosynthesizing, when they're, you know, they're producing oxygen when the sun comes on the plants, the oxygen has to come out somehow, and so we think that it comes out and makes bubbles and whistling sounds. But we haven't been able to actually do, you know, show that for sure. So we're really interested in looking at that as well.
It's strange because we've got this new app out, which is the Song Meter Touch, which is just a recording app where it records back in time. I was mentioned at the beginning, and you can plug other things into it. And I have a little garden pond, and I bought myself a little hydrophone, and I've been plopping it in the pond, and I'm getting water boatmen and lots of other things making strange sounds. But yeah, you get the plants, just like little factories all over the place. It's brilliant. It's really good fun.
Yeah.
So get yourself a cheap hydrophone and plug it into your phone. That's what you need to do if you want to carry on researching in that way. The impact of anthropogenic sound on birdsong is well documented, notwithstanding data gaps. Do you have any thoughts about the wider impacts of anthropogenic sound on insects?
Yeah, there's not much research at all on this topic. So I can think of a few examples where they show that you know, insects tend to respond to noise as well, and so in some cases it looks like they're increasing their activity in response to noise. So maybe they're responding to noise as if it were like a competition, so they have to get over the noise, or they perceive it as another individual, you know, that they have to compete with. But yeah, it's very interesting because we think about, I mean, how we impact our environment is you know, just with the noise, it's just overwhelming. It goes everywhere, underwater, in plants, and everywhere.
Yeah, the laws of physics underwater are different as well to above water, aren't they? Sound bounces off the surface and it travels faster and all that sort of stuff as well. Oh yeah. I've only just got my head around it on land, I think. More on tree hoppers. If I was standing among some tree hoppers, could I actually hear them? Or is the sound outside the limits of human hearing?
Yeah, no, you could. I mean, they're not audible without something to perceive vibration. So I know that the first people who worked on vibrations, they were able to you know, put the insects in little tubes and then put the tubes into their ear, and that's how they managed to find out that insects actually produce vibrations.
That's the 3 o'clock in the morning experiment, that one, isn't it? Someone's asked this question, and I don't know if it's a person that's here, but are you going to look at soil bioacoustics as well? I know there's been a lot of work being done on soil bioacoustics.
Yeah, it's I mean, it's fascinating. I would love to dive into that, but I'm not sure. This is not for a plan for the near future, but I know some of my colleagues are doing some wonderful work on it.
Yeah, contact microphones and burying them in buckets of soil and things like that. Yeah, it's good fun. This is quite an interesting one. How do you expect your work to guide conservation strategies?
So the last part I was talking about in my talk, the project, we are aiming to predict the distribution of acoustic groups with future environmental conditions. So we can, for example, assess the future, the effect of climate change, for example, or also change in land use. And so we can give information about what the sort of acoustic landscape will look like if those changes occur. And so we can give information to conservation managers to say, well, maybe it's a better idea if you don't do this, but do this instead, and things like that. So that's the project. That's the aim. The end aim.
Well, I think we've probably only got time for one more question, because I've overrun and not been a very good host. So does the tree hopper prefer any specific host plant while calling?
Yeah, so they're very specific to their host plants. So there are actually it's a complex of species. There are about 10 species, and they each have a specific host plant that they live on, and they have very low success reproducing on other plants than their own, and they don't survive well also because they can't, they won't be able to eat on those different host plants. So yeah,
So they're plant specific.
yeah.
Thank you so much. There's loads more questions, and I think if you've got a bit of time to go through the Q&A, it'd be really useful. But I can't thank you enough for that. That was really interesting, and I've learned so much today. It's been brilliant. But yeah, thank you very much for that. And yeah, I'm sure she'll be answering things in the chat, and loads of clappings going up again, because yes, they will do. All three speakers been brilliant. Thank you so much.
Yeah.